DNA manipulation, also known as genetic engineering or molecular biology techniques, involves the alteration or modification of an organism's genetic material, DNA, for various purposes, including research, medical applications, and biotechnology. Here is overview, principles and methods involved. 1. Isolation of DNA. DNA manipulation often starts with the extraction and purification of DNA from a source organism. Common methods include phenol chloroform extraction, column-based purification kits, or magnetic bead-based techniques. 2. Cutting and splicing DNA. Enzymes called restriction endonucleases are used to cut DNA at specific recognition sites. DNA ligase is used to join together DNA fragments by catalyzing the formation of phosphodiester bonds. 3. DNA amplification. Polymerase chain reaction, PCR, is a technique that allows the selective amplification of a specific DNA segment. It involves cycles of denaturation, annealing, and extension, using DNA polymerase. 4. Cloning. DNA fragments, genes, or entire genomes can be inserted into plasmids or other vectors and then introduced into host organisms to replicate and express the foreign DNA. 5. Sequencing. DNA sequencing methods, such as Sanger sequencing or next-generation sequencing, NGS, are used to determine the nucleotide sequence of DNA. 6. Gene editing. Technologies like CRISPR-Cas9 enable precise editing of DNA sequencing by introducing, deleting or modifying specific DNA segments. Advantages. 1. Medical advancement. DNA manipulation allowing the treatment of genetic disorders. 2. It enables the development of genetically modified organisms, GMOs, for the production of pharmaceuticals, vaccines, and therapeutic proteins. 3. Biotechnology. DNA manipulation plays a crucial role in biotechnology, including the production of genetically engineered crops with enhanced traits like disease resistance and increased yield. 4. Scientific research. It facilitates the study of gene function, regulation, and the role of specific genes in various biological processes. DNA manipulation aids in the creation of transgenic animals and organisms for research purposes. 5. Evolutionary studies. DNA manipulation allows the comparison of DNA sequences across species, contributing to our understanding of evolution and phylogenetics. Disadvantages. 1. Ethical concerns. Manipulating human DNA raises ethical dilemmas, such as the potential for designer babies and 2. Environmental concerns by release of genetically modified organisms into ecosystems. 3. Safety risks. The accidental release of genetically modified organisms can have unforeseen ecological impacts. 4. Causing unintended genetic changes and socio-economic issues. 5. Unintended consequences. Modifying genes can have unforeseen consequences on an organism's health or behavior, with potential long-term effects. 6. Altered genetic traits may disrupt ecosystems if released into the wild. DNA manipulation offers enormous potential but requires careful consideration of ethical, safety, and regulatory concerns to ensure responsible use and minimize risks.